All right, so today we are looking at the first animal kingdom specimen or representative, and this is the phylum Porifera. Now, you're going to hear me say this again and again, repeating myself as usual, but this phylum of Porifera probably could just as easily be classified as a protist. So the only reason I think that it still remains in the Animalia kingdom is because of tradition, is because this, it's been described that way for some time. But I mentioned last time that we have a protista issue. Let me pull up this other PowerPoint from last time. The protists. Let's see, where have protists? Last time I had on my list, I went all the way to here, and then I said we're going to cover these flagellata today, and that's what we're doing right now. And if we go to the very, very end of the picture, I don't have any specimens of these because they're like really rare and they're really delicate. Great to have some specimens of them. But basically, the flagellata is a phylum of protists. They're all ocean dwelling. They're all marine. Anyway, how they so this is, let me go ahead and just draw this. So you can and, I'm drawing it just so you can kind of get an idea of why we group these with the sponges. So this is the phylum flagellata. And this is in the kingdom Protista, which is the protozoan, right? Okay, so what we got going on here is we have a cell that has a, so here's the cell, the nucleus there, and it has like a collar on one end. It has like a kind of like lacy or like a sieve, like almost like a gauze like end to it. And then these live on stalks where they attach themselves to like a rock or organic material or whatever. And then there is a flagellum that emerges from here. So this is the flagellum. And then this part here is called the collar. And I think they call it the collar because it kind of resembles those like the Shakespeare frilly collars they wore, frilly cravats or whatever. It's kind of like one of those little frilly collars from like the Victorian era. I like to think of it like this. It's kind of like a badminton shuttlecock, right? These little badminton things. So I kind of, when I saw this, I stole this from my stepmother. Anyway, I kind of feel like, wow, that's a lot like a flagellate collar, and this is like the cell here. And if only there was a little flagellum here, that would be a really good example of what the flagellate is. So what they do is they whip around the flagellum. It's not used for locomotion because they're sedentary. They're attached to one spot, but instead they draw organic material and microscopic organisms through the collar. The bacteria and organic material attach to the collar, and then they ingest them into the cell. So that's kind of like their feeding collar, basically. It's not for locomotion, it's for feeding only. So anyway, that being said, the sponges basically have a feeding cell that really strongly resembles that same feature. So the choanoflagellate cell has this collar on it, the flagellum. The choanocytes, collar cells, that the sponge has for feeding, has a very, very similar structure. And so some time ago, probably in the early 1900s, it was pretty well acknowledged that sponges and these choanoflagellates are probably pretty closely related. But they kept the sponges in the animal kingdom because sponges are multicellular, whereas these choanoflagellates are single cellular or colonial. And that's one of the definitions of an animal, is animals, by definition, are multicellular organisms. There's no examples of a single cell animal. So that's where the difference lies. So, but just like the other protists that we saw, like the Cladophora versus the Chlorella, you could say that if we were to put the sponges into this phylum Choanoflagellata, I don't think very many biologists would cry about it. I think it's probably, they make a lot more sense. Even with DNA analysis has also confirmed that these are probably pretty closely related groups. It's probably the sponges are more related to this protist phylum than they are to any animal phylum. So just let that be known. That's kind of the idea. So anyway, now we're looking at the kingdom Animalia and the phylum is Orthra. Okay, so what's going on here is we have organisms that are multicellular, so they have a multicellular organization, but a key thing about these that you have to know for the test is they have no true tissues. And what that means is a sponge is basically an aggregation of cell types. There are no tissue layers like an endoderm or an ectoderm or a mesoderm. Those are the dermal layers that make up most animal structures. These don't have any kind of endoderm, ectoderm, or mesoderm. There's no epithelium, no muscle, nothing. It's basically just three basic cell types that kind of aggregate together to make up the organism. They are asymmetrical. And what that means is there is no head, no tail, no top, no bottom. A sponge basically is a cluster of cells that are, arranged for feeding in a certain way, but there's no anterior, posterior, left or right. It's just a ball of cells, basically, or a cluster of cells. So they have asymmetry, which no other animal has. All the other animals have some form of either radial or bilateral symmetry. Okay? And the third that's pretty cool, a pretty cool feature, or I think is they have totipotent cells. All the cells are totipotent. And what that means is totipotent means a cell that could give rise to any other cell type in the body. And you probably know that term as like a stem cell, right? Stem cells. You've heard that with regards to human health and medicine. What a stem cell is a cell that could be used to regenerate any cell type in the human body. Where that normally comes from is, for example, like nerve cells in our bodies can't regenerate. And there are basically no cells that can give rise to nerve cells once the nerve cells are established. So you know when you have nerve damage, it's like permanent, right? Totipotent cells means these are cells that could give rise to any other cell type in the sponge. Sponges don't have nerves, but basically, you could take, there's an experiment with you on this. You take a sponge like this, you can throw it in a blender, grind it up in a blender, and then you can take a little toothpick, smear a little sample of sponge smoothie onto a rock, and it'll grow a whole new sponge. So it's like basically you can take any of the cells of the sponge, separate it from the sponge, smear it out, and it'll grow another sponge. So it's basically kind of like a regeneration superstar. And it's actually the only animal that can do that.
No other animal has fully totipotent cells. Once you separate ectoderm and endoderm and mesoderm, usually those dermal layers can only replace that type of tissue, right? So you wouldn't be able to generate different type of tissue in any other animal. So, as such, they are an important research organism. So studying stem cells, sponges are kind of like a model organism for how that works, right? Okay, so let's go ahead and take a look at the body plan of the sponge, and we'll kind of get an idea of how they operate. My colors here. The body plan is kind of like a base, simplest body plan, which is called the asconoid body plan. It's like a vase-like plan. So if we had a sponge here, let's see, I'm trying to pick the colors here, some appropriate colors. Yeah, fine. We have kind of a vase-like body, like flower vase or something. Okay, and so how the body kind of works here is, let's see if I can do this right. We have an outer layer of what are called canapocytes. Is kind of the outer layer here. The inner layer is made up of melanocytes. Should have made my ostia after. Okay, so what we got going on here is we have the canapocytes on the outside. We have the choanocytes on the inside of the thing. And then some other features that you have to know are this cavity here, this cavity in the middle, is called the spongocoel. Spongocoel, C-O-E-L in Latin, means cavity. So that's a spongocoel, sponge cavity. And these openings here on the sides of the sponge, where the water is drawn in, are called ostia. And then this opening, the common opening of the cavity where the water is expelled, is called an osculum. Okay, so that's kind of like the basic overall sponge body. Kind of the sponge on the left is that kind of example. Let me show you kind of a exaggerated diagram here. I'm gonna go ahead and take like this little piece here and draw like an enlarged version of it, so you can like magnify it or zoom in on it. And the inner layer of cells are these choanocytes. So we have these kind of like badminton, shuttlecock, or birdie cells that happen here. This, okay? And then these will all have their okay, and then these cells have their flagellae facing the inside of the spongocoel. So these are choanocytes. All right, and they are lining the inside of the sponge cavity and waving their little flagellae around. And what that does is it draws water in from the ostia. Water goes in where the arrows are. It circulates in the spongocoel. They collect food particles from the water, like organic material or microbes like bacteria and small yeast cells and smaller protists, and ingest them. And so these are the choanocytes are the feeding cells, basically. They set up the circulation and they establish the feeding with the feeding food brought in. Okay, and then on the outside of the body, we have these canacocytes that are kind of like flattened cells. These are canacocytes. And they're essentially making the body, the outside body of the sponge. This middle area here is called the mesohyle. So meso means middle, that's easy to remember. And in that mesohyle, there is a third cell type that happens or occurs, and this is called an amoebocyte. And what the amoebocytes do is they look like amoeba, is they will collect food from the choanocytes and then migrate across and deliver food to the canacocytes. They're kind of like the DoorDash of the sponge, right? So they're basically responsible for kind of transferring food around the sponge. These are called amoebocytes. So that's it. There's only 3 cell types, or there's a couple more, but I'm not going to worry about it. But these are the 3 main cell types for how it functions as a multi-cell organism. And why it's called multicellular and not colonial is, in a colonial organism, each cell is responsible for its own feeding and reproduction and everything else. Here we have a division of labor. It's like the feeding cells, the distribution cells, and then kind of the skeletal cells, the body cells. And they cooperate and work together as a unit. Okay, in this mesohyle, there can also be a number of different skeletal features. And the one I'm going to show you here is spicules, is a unique structure found in sponges. Spicules are these sharp, almost like barbed wire or tack-like features. Usually they're kind of like shaped like harpoons or broken glass or whatever. But these spicules are defensive structures. Think of like barbed wire. If you were to wrap food in barbed wire, it'd be hard to eat. And so very few organisms eat sponges because these spicules end up being like eating broken glass, right? And that being said, the classification of sponges is based on what type of spicules they have. Okay, there are 3 basic sponge body types. This is kind of an archaic way to describe it. So I'm not going to test you on these names because I don't think it's necessary. They're kind of old school. But anyway, the smaller sponges tend to have a real simple body plan, like a cup-like body plan, like a vase. So that the one I've drawn here is called an asconoid body plan, which is like just a simple cup. As the sponges get larger, to increase the feeding surface area, they end up making pockets or side pockets on the spongiocele. And the largest of sponges are Euspongia, is kind of an overwhelming collection of pockets. So what we're seeing here is actually hundreds of individual spongioceles all clustered together. This is a leuconoid body plan where all those little pockets are little side pockets of a spongiocele. Most of the sponges that we're going to look at, though, are very, very small. I have these, let's see if I can find them. Here we go. Yeah, these are kind of like the ones that we're looking at here. Leucoselenia, and this is just for you to look at in the jar. On the exam, it'll be on a slide. So, but this is so you can see the whole organism. They're each individually very, very small, maybe like half a centimeter in length, and then Grantia is maybe about 2 centimeters in length. But basically, as they go from a half a centimeter to 2 centimeters, they have to go from like simple cup, maybe a more complicated cup, and then as they go to like 10 centimeters, like this size, they have to go to like this porous kind of gallery cup. Okay. So that being said, let's go ahead and talk about the 3 classes of sponges that we're going to look at. So the class level is the next level after phylum, and we have, well, let's go ahead and pull it up here. Calcarea. Class Hexactinella. And then class Desmos. All right, so there's our 3 sponge classes. The Calcarea are called the Calca sponges. That's in quotes because what's going on here? Okay, these have calcium carbonate spicules. So the spicules or spines in the Calcarea are made of calcium carbonate. How they test this is they use hydrochloric acid, and they'll drip hydrochloric acid onto a sponge specimen, and if the spicules dissolve because of the calcium carbonate, they know it's a calca sponge. So the calca spongia has, we have 2 different ones to look at. We have leucoselenia. And we have Grantia. Again, I'm not going to ask you those names, but very well on a microscope slide, I'll leave the label uncovered, and if it says leucoselenia, it gives you a huge hint as to what it is, right? I mean, you'll know by looking at it, but it'll be the additional resource for you to look at. Okay, leucoselenia. We'll come back to the hexactinella and Desmos spongia in a second here. Leucoselenia basically looks like this. It's like a bunch of little vases or fingers on a sedentary sponge. Grantia kind of looks like this. It's like a fuzzy sponge because it has all kinds of side pockets. And let's see here. Leucoselenia is here. This is basically a whole sponge kind of on a microscope slide. And it looks something like this. It's kind of greenish. But leucoselenia is pretty much this model of a sponge. What you're going to see is something like this. Sometimes you'll see like 2 in one on one microscope slide, and then you'll see like a tear where they ripped it off of the main colony. Okay, so that's just kind of like a broken bottom. But what's going on here is you'll see these are covered in spicules. You'll see it looks like a cactus almost, because these little spiky spicules are all sticking off. So the things to note about the leucoselenia on the slide there is these calcium carbonate spicules. You wouldn't know the calcium carbonate without testing. But if it's leucoselenia, you know it's a calcium sponge, so I think that's the way it is. And then the other question I like to ask is, This is the osculum. This is where the water exits the body. Water is brought in the ostia, circulates around, and then is pushed out in the osculum. So you can see the osculum on these little sponges. So these are good questions to ask for a test. The questions would be the class name of the sponge, Lucaslinia, the phylum of the sponge, Porifera, what are the spicules made of on the sponge, calcium carbonate, and what is the little opening at the end of the sponge there. I almost said top, but there's no orientation right at the end. And it is the osculum. Okay, in the Grantia slides, this is so you can see the choanocytes. So I have Grantia choanocytes. So what you're going to do is you're going to look at a cross-section of the sponge. And so these Grantia choanocyte slides are cross-sections or long sections. And I think what we're looking at for the most part is cross-sections. So one way to tell is on these slides, they'll say CS or LS. And so if you see CS, it means cross section. Okay? If you see LS, it means long section. Either one of these is okay. The cross section is better. And on the exam, I will have the cross section presented. That being said, if you look at long section, you can see the parts that I'm talking about. It just won't look like this because we're not getting kind of a sample across the middle of the sponge. You're getting a sample down the length of the sponge. So I think the cross section is better, but neither of them are wrong. They're both pretty good. So I'd say check the cross section out. That's what I'm drawing here. And the cross section basically is going to show you essentially something like this. The Grantia cross section is going to look like, draw that up there. What's going to look like is this. Okay, and you're going to see like some mesohyal in here. The other cool thing you're going to see in this cross section is you're going to see some amoebocytes. Okay, but in that cross section, you're probably not going to see pinacocytes. The problem with pinacocytes is they're so thin that when they do the cross section, they're not really visible and they get kind of torn away. But just for your perspective, the pinacocytes would be kind of surrounding the outside. Maybe they're there, I just can't tell them from the other stuff that's going on there. Okay, so here's what I'd be asking you here is the choanocytes. The choanocytes basically are going to look like this. On this inner layer here, this is where the choanocytes are lining the inside of the spongiocele. And I'm just going to go ahead and point to this diagram, because that's what you're going to basically see, something like that. It's not going to be that detailed, but it'll be what you're looking at. So you can see on this diagram, you can see lining the inside, you can see these kind of long, kind of trumpet-like cells, and sometimes you can see flagellae present. And those are the choanocytes. Looking for the amoebocytes, you can kind of see there's one there, there's one there, there's like one there, the amoeba sites, they look like little amoeba, and so they're pretty obvious. Now, whether these outside parts are pinacocytes or not, I don't know. But the two questions that I would ask you would be the choanocyte layer. And I like to put a pointer or something on an amoeba site. There. So make sure you see that, because that's what the test is going to have on it. I'll have that slide on the microscope, and I'll have probably something pointing out the choanocyte, something pointing out the amoeba site, and you'll have to tell me what those are. Okay? That's a grand joke. All right, that's what the purpose of those two slides are there. All right. I think that would be good. I think the spicules you'll see on the with the selenia. Okay, so Hexacamelida. This one I only have one specimen of it right here, and this is really cool. This is a really beautiful sponge. Go ahead and this one we'll leave it in the box up here. So you can take a picture of it up here. So don't take it back to your desk because it's very fragile. And I will put it into the box like this so it's kind of propped up. But this is called a glass sponge. It is it is found in like the Western Pacific, like kind of in the off the coast of Japan and kind of south of New Guinea. But these are called glass sponges. The genus is Euplectella. But again, I'm not going to ask the genus. And these have silica spicules. So SiO2 spicules, which is why they're called the glass sponge. They're all marine. The sponge body is kind of that same kind of sponge body as Grantia has, cycanoid. And the fun, and how they tell this is when they drop hydrochloric acid on those spicules, they don't dissolve because silica is resistant to acids. There's a fun story in here that, I don't know, I'm not going to ask you the story in the test, but this is a fun story. If you look inside of this sponge, there is, so for one, this sponge traditionally is given as a kind of a ceremonial wedding gift, like in Japan, like in traditional Japanese weddings. They'll give this sponge as like a ceremonial wedding gift. For one, it's very ornate, it looks like lace, like an elaborate crystal. But for two, if you look inside, you'll see two little shrimp claws that occur inside of this sponge. There's two little shrimp claws. So what happens is, early on in the sponge's development, it will be colonized by larval shrimp, a number of larval shrimp. Anyway, in the end, I don't know if they compete or have death panels or whatever, but in the end, it's colonized by a male and a female of this shrimp species. It's very specific. It's very unique species of shrimp that specialize in colonizing these glass sponges. So the shrimp larvae get in through the ostia, and then when they mature, they're too big to get out, right? So they're essentially stuck in this spongocoel. And but what they do is they essentially feed on, they clean the inside of the sponge, they feed on the excess food resources of the sponge, and they mate and they have babies. And the little ball you see in here is like a ball of their poop, or their waste. So they kind of collect their waste into a little latrine and put it on one side. They're basically living the happy, blissful prison of marriage, right? So it's they're all stuck in there. that's why this is given as a ceremonial wedding gift to represent, the eternal prison of marriage in a sponge. There you go. So, won't ask it on test, but you'll see that little clause in there. It's kind of interesting. But there is a description here on the sponge of where it's found and things about it. So you might want to get a picture of that for your reference. Okay. All right, the Desmospongia. Desmospongia are the most diverse of sponges, and it includes the only freshwater species that exists. There's only one genus of freshwater sponge that occurs. We have these in Shasta County. I found them in Whiskeytown Lake. They usually kind of look like a yellow scum on the rock, but it's actually a sponge. But these can have calciferous or silica spicules. They have spicules, possibly, maybe not. But the main feature of these is they have a unique protein fiber called spongin. And the spongin fibers are the fibers that make up the kind of spongy, real elastic, what we think of when we think of sponge. That's all sponge fibers making up your skeleton. So, I would say that in this mesohyle, not only do you have spicules, but you can also have sponges. And that is also a skeletal feature that only occurs in the Desmospongia. Okay? So these are called the commercial sponges. And I think I have a slide of sponge fibers. Yeah. Here is a slide that basically is a microscope section of a commercial sponge. It basically looks like spaghetti, like blue spaghetti on the slide because they stained it with a blue stain. So it kind of looks like spaghetti. So when you look at a slide, people usually raise their hand and say, all I see is like, two, little lines or what looks like spaghetti. And that's just the sponge fibers. All the tissues have been taken away, all the cells, the melanocytes and pinacocytes have been dissolved and got rid of, and the only thing there remaining is just the fibers. The sponge body here is the leuconoid body plan. Because of the size, they need to have the increased surface area for feeding. But there's quite a bit of diversity. We have 3 different types here. There's kind of maybe encrusting sponges like this that are kind of hard. You wouldn't want to bathe with this because it would scratch you. Unless these have spicules in them, you can kind of feel them. If you rub this on your skin, it kind of scratches your skin with the spicules. But you know that bath sponges are still available. You can buy sponges, like real sponges, and it's a huge commercial industry still. It's probably one of the oldest commercial industries in human history. There still is a commercial sponge industry in like the South Pacific, off of New Guinea, and the Philippines, and in the Mediterranean. There's a sponge industry where they dive for sponges. And you can go, I think you can buy these like at the hardware store for painting. You dip it in paint and you can put textures of paint on walls, or you can buy them at Michael's in the art supplies section for painting. But they're pretty expensive. Like a sponge like this would be like $20 or $30. Maybe even more now, I don't know. And of course we have alternatives now. The sponges that you buy like for cleaning your dishes, etc. This is made of sponge rubber, right? So it's basically a rubber plastic compound. That's not true sponges. But of course the name lives on, so we have to kind of honor that. Okay, finally, I told you this would be kind of a shortish summary here. You should be aware that sponges are really important in the study of medicine. Namely, they're looking at sponges as a way to come up with anti-cancer drugs or anti-tumor drugs. One thing that sponges are good at is because they stick themselves to a rock and live there their whole life, they have to kind of defend that habitat from other species or other organisms. So sponges produce all kinds of chemicals that are called allopathic chemicals that will prevent other organisms from growing them over. Many of these chemicals operate by interfering with cell division. So they interfere with the cell division pathways of the competing organisms, or they will alter their cell function, like they'll neutralize the mitochondria of competing cells. Or they have anti-inflammatory, where they basically slow the growth of the cells around them. So these sponges are being looked at as possible chemicals that could be altered or modified to be used as anti-inflammatory drugs or anti-tumor drugs, etc. So it's a cool area. If you're interested in medicine, but you don't want to become a physician, you want to become like a MD, PhD type researcher, this is a really, really hot area to look at for research. Okay. So I don't have any questions on that, but I think it's just a cool way to kind of appreciate what the sponges are all about, right? Okay, so that is it for lab here. I have a little list of terms here that you should be aware of what these are. These will be like answers to test questions in addition to class names. Any questions? All right, so that's laid out kind of in sequence, and this PowerPoint is available on Canvas, so you have that available to you as well. And if you need to see any specific proto slides from last time, let me know. I think what I might do is, I might take these slides, because we have a full class here. I think what I'll do is I'll put them over there on that side thing over there. I'll lay them out kind of in sequence. And if there's any proto slides that you need to see, because you didn't see them last time, this is kind of like the last chance. And we'll have them here, but next week we're going to get into more stuff. And it's just going to kind of become overwhelming. So you'll find out that we will accumulate a lot of information leading up to that September 12th, September 19th. All right, there you go. Have at it.
A little fast to get going.
Paparazzi. The zoo paparazzi. Okay, so these, be careful of these. We have
I was gonna double
they're kind of perched here tenuously, but these are the slides from last time. Yes, they don't need them when that's part of the thing. That's a lot of calcium and calories to make those.